I want to welcome you all. Most of you were here yesterday, so I won't bother giving any real speeches. But again, I want to thank the different organizations on campus that are sponsoring these workshops. They are the Interdisciplinary Data Sciences Consortium, the Integrative Biology Department, the College of Arts and Sciences, and Research One. All have contributed in various ways. And I'll add one other thing, that we are expecting to hold more workshops in the future, and we would really appreciate your feedback from these. So if you have either suggestions for topics or comments about what worked and what didn't, or whatever, let me know or send me email or anything like that, and we'd really find it quite useful. We missed an holiday. 2 days ago, we discovered, my wife discovered this last night. She said, it's World Statistics Day. And, well, actually, the previous day had been. So now we're 2 days late. So, you know, eat a cookie, and we'll break out the cocktails later? I don't know. I had no idea there was such a thing as World Statistics Day. Next year we'll have a big party. Okay. So, if I can find the right, there we are. So, I want to talk with you about a topic near to my heart. Censored data and truncated data. They're not quite the same thing. They bear a number of similarities and a number of conceptual issues, but they're not quite the same thing. I will spend the overwhelming majority of time talking about censored data, just because I need to do one of these topics justice, and that's my choice as to which one I want to spend more time on. These are the words are a bit mysterious, as names like this always are. What does censored data mean? In fact, most people actually assume that that's the NSA is knows every keystroke you're making, but they're censoring your availability, your access to that data. That's not what we're talking about. We're not talking about governments restricting your ability to get data. That's a different, I suppose one could use the same English words, but it's an utterly different meaning. Censored data and truncated data are things that actually we encounter in an enormous number of contexts. I first encountered them when I was doing, in graduate school, as part of survival problems, and they're very characteristics of survival problems. Just to give you the classic example, in a medical study, let's imagine we have a drug, and we have 100 patients, 50 of them getting the chemotherapy drug, and 50 of them getting placebo. And we want to know whether the drug is any good. And so of those 50 getting the chemotherapy drug, 5 move away after we've given them, and we're recording the time until they have another symptom, or the time until death. And so of those 50, 5 of them report back to us for a while, and we know they're doing okay, and then they stop coming. Maybe they moved to a different city, maybe they got tired of being part of our study, whatever, we don't know. 6 of them have step off the curb and are run over by buses. And the remaining 39, we have data on. So the problem is those 11 people that we have partial data on, we know they didn't have any symptoms until they were run over by a bus or until they stopped coming back to visit us. And the temptation there is to throw the data away. That's what a lot of people do. They say, well, we don't know, so we have to ignore those data. There are other ways that people address it, and most of them, I'll just begin by saying something that, as directly as I can, most of the other ways, and most of the ways that you think of to do it, are bad. Bad as in evil, don't do it. Okay? I don't want you to think about doing these things. So there are ways to address these problems. Because I should say, theory for handling censored data problems is behind theory for ordinary problems. You know, there are these things related to linear regression models and its cousins like analysis of variance. Those are very far advanced, partly because they've been known since the 30s probably, and partly because, well, they're relatively simple problems. These are harder problems, so of course there are things that you may not be able to address properly with these methods. Okay. So first I want to give you an introduction to censored data, and I sort of did. I'm a population ecologist, so those are the examples I tend to think of, but there is nothing particularly remarkable about ecology or biology in these sorts of problems. So the basic point is our data are often complicated. We often have situations like the one I just described where for some things we have partial data, and I'll talk in a minute about in fact, the principal examples I'll use are a bit more insidious than the examples of some patients dying of causes external to the study, like being run over by buses. The problem is these complications in our data can really affect our estimates of the various quantities we want to estimate. And we do have methods to address these. One of the questions, of course, is when do you use these methods? So without going much further into it, let me just introduce the problem. If I have 10,000 data points and one of them is a case like, you know, a patient getting run over by a bus, do I need to worry about this? And of course the answer is, I don't know. There is no good answer. The practical answer is you may not have to. It may not affect your estimate much. Just as if you're doing a linear regression, you know, a bit of heteroscedasticity in the residuals may not be anything to worry about much. There is no such thing as perfect normality, or to put it another way, you could never demonstrate it anyway. And so, you know, I'm going to tell you, don't do certain things, but it's also important not to treat these things as though you're a religious fundamentalist and, you know, we must do things, this is the only correct way. Not always true. Okay. So here are some examples. Let's imagine you're wondering whether the water someplace is safe. So scientists go out and they take samples. We have a colleague, in fact, our department chair does just this thing. Her lab takes samples of the water, and they'll measure the density of fecal coliform bacteria in the water as an indicator of how polluted it is. Here's the problem. She can only measure densities of bacteria over some quantity. If they're very sparse, if they're present but very sparse, they won't show up in the measurements. So it's the same problem of, nobody here has this problem, but if you had a scale that was, you know the scales when they measure, when you move in a moving van, they weigh the truck empty, and then they load your stuff onto it, and then they weigh it again. I'm going to guess that if you step on that scale, it will report that you weigh 0. You know, it just doesn't have the sensitivity to measure one of us. In both of those sorts of cases, that's censored data. We know that there's a data point there. We know that my colleague measuring her bacteria, she's taken a measurement. We know that the density of bacteria is not greater than whatever the sensitivity she can measure, but we know it's between that sensitivity and 0, inclusive. Okay. Similarly, you know, stepping on the scale, the true weight of me is between 0 and whatever the smallest amount that scale can weigh. Okay, there's a similar problem, another ecological example. People sometimes do things like they use satellite data, and they'll try to measure the density of, here I say, some species. Imagine you're trying to ask how dense are the trees in a region, because you're interested in, well, in forestry, or perhaps in some hydrological problem where the amount of vegetation is really important. If the trees are very, very sparse, you will not be able to measure them using that satellite data, because, of course, it has the images that the satellites get are averaging over for each pixel. They're taking some measurement and they're taking averages over those pixels, and so you can have a non-zero density of trees that you can't measure. And this happens all the time in data of that sort. Imagine you're trying to study survival time, and this is sort of the example I gave of the patients. But I'm going to study survival time of some fish. There are a lot of reasons people do this sort of thing. And I'm going to keep them in the lab. I have funding for 3 years. At the end of 3 years of my 200 fish, 5 of them are still alive. What do I do with those data? Do I say they lived 3 years? This is what some people do. Some people say, well, it's infinite. It's infinite. Well, it's not infinite. they will die. And it's certainly not something like the mean of infinity and 3 years, which is still infinity. I've seen people try that. So sometimes people will say, well, I'll pick the largest number that I think they could possibly live. I think they might live 5 years. And you can see that doing all of these things present problems in that you can potentially really introduce serious biases into your estimation, and you can get, even worse, you can get what statisticians call inconsistent estimates. Well, and I'll talk about that a little bit. So where does the term censorship come from? I have no idea. I've tried to find out. I need to talk to some, historian of statistics. Every time I have looked it up, what I get is a lot of information on the dictatorship in Argentina censoring people's access to data, and I just, okay, forget it. I'm not going to waste more time with Google on this. We do this a lot. You know this expression, when all you have is a hammer, everything looks like a nail? We do this a lot. We say, well, I know how to, I have a toolkit. I know how to do this, and this other thing. So I can twist my data in some fashion and make it, I can bend it to my will and make it conform to the assumptions of some statistics, some statistical method, sort of. And then I can use the statistic. And sometimes that's fine. I mean, to give a simple example, sometimes you have data where, if you log transform it, it becomes approximately normal, the model makes sense, and that's fine. That's not a problem. It just means you needed to think about it on a different scale. But sometimes it's a really bad idea. And what I want to argue is, stop doing that when you have censored data. Okay. So the most common method people use with censored data, the most common wrong method, is what I'll call ad hoc substitutions. So one approach, I should say, is to just delete the data. I have these, right, I don't know how long the fish will live, so I will ignore those fish. And if you think about it, if you're trying to say describe a survival curve, you ignore the most long-lived individuals and throw those data out. That's a bizarre way to do things, and a severely biased way potentially. And the other common thing that people do is ad hoc substitutions. They do things like what I just said. They'll set a value. They'll take a value and arbitrarily substitute it. So typically, in a laboratory setting, for example, my machine can only measure the concentration of, I'll say, a pollutant in water as low as 100 units, whatever they are. And so I know that I know that I have a sample that registers 0. So I will arbitrarily call it 100, the minimum I can, or 99 or something like that. Or I'll say, well, better than 100 is the mean of 100 and 0. It actually usually is better than 100, but so I'll call it 50. And go with that. That's actually a very common procedure, especially in that sort of setting where you have some assay with a lower limit of sensitivity. People often say, well, I know it's probably not 0. I'll call it the mean of sensitivity and 0. I'm sorry, I should have introduced LOD, limit of detection. And that could be either a lower limit or an upper limit. You may not be able to measure things more than something. If I eat a lot of cookies and I step on my bathroom scale, the dial will go around a couple of times and come to a stop at some point, and all I know is I weigh at least 350 pounds or something like that. I don't know. It's 350 or more pounds. So we can have a limit of detection that's lower or higher than our data. So why is deleting data bad? Why are ad hoc substitutions bad? I mean, I've sort of mentioned in general these can introduce biases, but the more important point is that ad hoc substitutions often introduce structure into the data set that isn't in the actual sampled population. And they can, and I'll show you some examples of that. That's actually what the scripts we have do. They also make estimates inconsistent, and we'll talk about this more. Inconsistency means, so if you have a biased estimator, it means that, let's say it's estimating low. But you know, bias isn't always so bad. If I get a big enough sample, I'll still get a good estimate. An inconsistent estimator, if I have an infinitely large sample, there is no guarantee that it's actually converging to the true population mean. Is that a fair enough sort of informal definition of inconsistency, Ram? Okay. By the way, I didn't say, interrupt me at any time if you have questions or. Okay. So let me give you a taste of some better approaches to handling this problem. So this involves using R a little bit. Let me find the, let's see. So there are several R scripts that I uploaded. The one that we're actually going to use is this one. It's the one that looks like we should use it. USF 2015 workshop, my initials, blah, 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 .r. Okay. And all the others are called by this. So all we're going to do here is, so first we need to set a couple of options. They're no big deal. And call some, whoops, load some libraries. And I just, as a matter of course, my norm is load all your libraries first in a script, even if you're not going to use them for a long time, just so that you know where they are. Okay. So I forget, it's either all or almost all of the data that we'll talk about today are simulated. And there's a real advantage to using simulated data for things like this, because we know what the right answer is. So what we can do, for example, is simulate a distribution. Let's say a normal, I'll take 1,000 normally distributed data points, and I know what the mean is, and I can chop one end of that distribution off and ask, well, what happens when we analyze it this way or that way? I know the right answer. So that's the reason for approaching this way. So what I'm going to assume with these data is a log normal distribution. And I'm going to assume that there's a single limit of detection. You know, you could have a limit of detection on the left and on the right. A scale is a perfect example. My bathroom scale can't measure the weight of a feather, and it can't measure the weight of an elephant. But we'll just keep it simple. I'll assume just one limit of detection, and I'll arbitrarily set it at a value of 0.5, whatever those units are. And we'll use some ad hoc methods for dealing with, that's called left censorship because we've chopped the left end off. So I'll set some values to the limit of detection itself. I'll do it with half the limit of detection, and I'll do it at 0. So the next line here just calls a little R script that generates the simulated data and does that simulation. And so the idea here is you can, at your convenience later, if you want to see how I actually did that, you can look at it. But it's sort of boring to actually run through the details here. So, but here's an example. So I've done that simulation, and the data set is called example 1. And here are some of the data values. And I'm sorry, I should bring this up a little so that you can see what the... So here are in the first column, there are the simulated data values, y. And these are the cases where I've, if it's less than half, I set it to half. It's y censored at the limit of detection. So let's see, there are no, this one. This one is 0.45, and it's set to 0.5, I'm sorry. All the others are, this one is also 0.19, set to 0.5. Okay. This is, we set it to half that if it's below the limit of detection. So that one's 0.25. Here are the cases where we set it to 0. And this is just an indicator, true or false, as to whether it's a censored value. And we know it's censored because we know it's below our level of detection, our limit of detection. Okay. So that little script, that's all it does, is it generates that data set. And I had a slide here that said this. By the way, just a complete aside, for if you're using random numbers, generating random numbers in R, there's a wonderful trick that you can use that is sometimes a good thing, not always. You can guarantee that your random numbers aren't random. Of course, you know that random number generators aren't really random, right? Okay. They can't be. How would you do that? And that's, so, it's using an algorithm, something highly nonlinear, to generate the numbers. But you can guarantee that, so when you want to do something like a demonstration, that they're the same every time. So you notice that the values on this slide are exactly the same as the values that I just generated in R, which seems like magic. That shouldn't be if it was really random, right? The command is set.seed, and then you give it a number. So if it's the same number, if I say set.seed 78, and I generate some random numbers, and I do lots of things, and then I do that again, I'll get exactly the same set of random numbers, which says they're not random.
Yeah.
Okay. I'm going over this bit kind of fast. Any questions? Okay. So here are some descriptive statistics, and this is all in that main. These commands are all in that main script, and the whole point is just to get a table of medians, means, and standard deviations of those different ways we're estimating y. So, the true median is 1.03, and it turns out, at least in this case, we always get the right answer for the median. The true mean is 1.67, and you'll see that these other things, it shouldn't be too surprising. In fact, it would have to be, wouldn't it? we've changed the estimates of the mean by doing these other manipulations. And we've also changed the estimates of the standard deviation. That's not real surprising either. And, you know, do those differences matter? Well, this is a simulation. Who knows? But it should be clear they might. And that's the real point here. They might. We sometimes have big effects, and I'll show you some cases. But it's not just on our estimates of the mean and median or the standard deviation or any other quantity. It's because we introduce, as I said, data structure, and I'll show you that shortly. So there's some better approaches than just making ad hoc substitutions. So one of the best approaches is what's called the Kaplan-Meier approaches. And some of you may know this term. Kaplan-Meier is the basic approach that's used in non-parametric survival modeling. And it's what you're asking in each case is what's the cumulative distribution of data. So for a survival model, and this is where how I came to this stuff, you're usually asking in one way or another. You're trying to come up with, at the very least, a description of time and survival, the fraction surviving, so it starts at 1 and it may go to 0. And you're trying to say, well, there's a curve that looks like something. It's obviously going to be non-increasing. And Kaplan-Meier method, it doesn't it really generates steps because you're estimating it every time somebody dies. So some of the steps might be really long, but it's the method that gives you a description like that. Those are supposed to be flat, but I can't draw. And this actually, so in survival problems, we confront this all the time because, as I said, you're trying to follow up individuals all the time, over time, and sometimes they disappear for various reasons, or you accidentally, in my studies, you know, we accidentally kill some of our study organisms. Too bad. Okay, the people who actually figured this out were astronomers. Astronomers are out there, they're looking at how many stars they see in regions, and they realize that they can't be seeing them all. Some of them are too dim or too distant. Or of course, there's true error in that if you imagine astronomers of the old days who were actually looking through a telescope with their eye and count, they would actually sometimes make mistakes, of course. And the machinery that does this now makes mistakes. So astronomers just asked, essentially, well, what's the cumulative distribution of the number of stars, and what does that tell us about the numbers that are missing? And they just invert the survival problem and approach it that way. So you can use a Kaplan-Meier approach. There's an approach that's got this ungainly name, robust regression on order statistics. And what it amounts to is this. So in a salud. So let's assume that we have a normally distributed data. So if you take the probability plot, the QQ plot, some of you might know it, if it's something close to linear, you can fit a regression to it, a linear regression. And it turns out that the intercept of that regression will estimate the mean for the whole distribution, and the slope will estimate the standard deviation of the distribution. So this is more general than just for normal distributions. You can actually use this for other distributions if they're in what's called the location scale family of distributions, which many of the distributions we use commonly are. But so it generalizes. If the probability plot of that distribution is close enough to linear, the intercept and slope estimate the location and scale parameters of those distributions. So this just seems offensive to when you first read about it. It seems like cheating, because you're estimating data that you don't have. When I first read about this, actually I thought it was clever, but I'm used to this sort of idea now. It reminds me of when I first heard about resampling methods, bootstrapping, for example. How can you make up data and then use that to estimate? How can you do this? You can get something from nothing. It's just wrong. Yeah, as I say here, it seems immoral. And the fact is, both of these sorts of approaches are using statistical theory to do this. They're just not doing it in the simple way that we're used to. If you're bootstrapping, you're not estimating the mean just by adding up all the numbers and then dividing. You're doing something else additionally. So I'll just assert that this is okay. And there's a body of literature that supports me. And proceed with that. So that's what regression on order statistics does. The robust approach takes it a little bit further, and what it does is it uses that regression model to impute the missing values, and I'll come back to the word impute in a second. And then it uses the pseudo data to estimate the mean and standard deviation. So I cannot give a talk about imputation. But imputation is, so it's more or less this approach. I have this intercept and slope, and I know something about where my data values are, and I say, well, okay, I'm going to simulate from that model what the data are. And you can do this repeatedly, for example. It's robust precisely in the sense that it's the regression on order statistics works, but it can be sensitive to details of your estimates. The robust approach, it works better, let me put it that way. It's less sensitive. If you want to know more about data imputation, there's a fine introduction to it with working our code. In the statistics book, the Statistics for Ecologists book, that what's it called? Ecological Statistics, that Simonetta and I and our friend Vinny Sosa edited, and I'll give you the reference later. There's a chapter on it by Shinichi Nakagawa, who goes over it, I think, in pretty clear language. Not for this kind of problem particularly, but for more general problems. So another approach is a maximum likelihood sort of approach. There's a regression model that we'll talk about in a minute that we can use to estimate these quantities as well. So there's a library. There's a guy named Dennis. Starts with an H. Hellcell, I'll think of it, who's sort of the guy who writes about censored data models, and especially a lot of his work, he's a statistician, a lot of his work has been with things like sampling of environmental pollutants and dealing with these problems of how do we do this. So he's the developer of this library, Nata, it handles a number of the sorts of censored data problems. So just to begin, back to that data set we simulated, I'm using I've loaded the Nata library here, and there's a command in it, senstats, the censored estimates of these statistics for that data set. And TF that's the name of, you remember I had a column that said true or false? That's the name of that column, so that the routine knows whether this is a censored data value or not. And now I have estimates of the median, mean, and standard deviation using the Kaplan-Meier, the robust regression on order statistics, and there's a maximum likelihood approach here. And you can see, well, now the medians actually differ from each other. The means are still different. They're maybe less different. And the standard deviations are still different. They all get sort of the same number. None of this is remarkably different. That's not always the case. Maximum likelihood approaches will not work as well if you have small data sets. And Kaplan-Meier won't work. This says will work poorly if more than half the data are censored. I think it won't work at all, if I remember correctly. It will certainly, you should certainly not trust it. But if you think about it, if you don't know the value of more than half your data, and you're actually trying to estimate the mean, and all you know is that they're smaller than something or larger, obviously you're in trouble. And maybe you're asking the wrong question. If there's that many data points that are censored, there's a problem there. Yeah, exactly. Well, you're using the wrong instrument, or maybe you just need to ask the question: is it less than something or greater than some value?
So can I ask you a question? So it seems like you must assume, even using this ROS approach, you're using the cumulative distribution of slope and its intercept to estimate this data that's at the lower end of your cumulative distribution. So you have to assume something about the shape of that cumulative distribution at the very low end when you don't have data at that end. You
have to assume that it's from the same distribution, because this only works if the probability plot is close to linear. So then you can, it's close to linear, so I can fit a linear model to it. And what you have to assume is those points that I only know an inequality for, it's less than something, are truly from the same distribution. If there's some other process that's making them censored, oh yeah, this will give you the wrong answer, absolutely. And I don't know of any methods that won't, because well, it's an interesting question.
Well, just thinking about a normal distribution plot, you could estimate the tail as linear here, the center area is linear here, right? But maybe all your data are in this region, okay?
Right.
And now you're saying, okay, maybe do you have data in the region of the plot? Do you actually see that data in the region of the plot where it's and it's really not linear either?
Yeah, but we're talking about we're talking about the QQ plot, not the probability function, not the distribution. So I can't remember whether I have a slide. I can't remember whether I have a slide of one. Let me quickly see if I do. Yes. Okay. No, I'm sorry, that's not a probability plot. No. Okay. So this is. Sorry, everybody.
I know.
Does ML even make sense in a censored data?
Pardon?
ML even makes sense in a censored data? You have to use EM algorithm to do that, right?
I'm not sure I follow.
Maximum likelihood estimate.
Yes.
Will not make sense in a censored data.
They can. They can. So essentially what you're doing is modeling. You have a two-part likelihood function. So you're sort of saying the likelihood is this. It looks like this if it's uncensored, and it looks like something else if it's censored. So typically, and again, I can't remember whether I have a slide that does this, that shows you what it looks like. So for okay, so this is a okay, that slide gives us something of what it's not the likelihood, but for this case, for example, we're going to be using a regression model where you say, all right, so we're greater than the limit. and that's our model. And this is what we're modeling what's observed here. What we observed was the limit. And from that, we can look at the expectation of the entire.
Yeah, but this one assumes that you have a cutoff.
Yes.
Only that place.
Right. Yes. So it gets more complicated, but so it's certainly possible that for my observation of you, the cutoff is one place, and the observation of Christina, the cutoff is someplace else. That happens all the time.
No, but even in your fish example, if you go
Yes.
back, let's say 15 fishes have been gone before 3
Right.
years, but 5 fishes which living,
Right.
at that point your C is basically going to be 5, but what happens to the previous values which already the data is gone?
Right. So essentially, what we will have is the case where, right, so some individuals are censored at the end of your study because you're ending your study, and you just have to say, I don't know. It's greater than this. Some are censored because you were taking a measure on them and you a measurement of them and you accidentally killed them. So you have to say, oh, well, I know that they survived up until this point, but I really have to take them out of the study at this point. And I can't call that just a death. It's not a death that's meaningful in terms of the study. And some are censored for, there are a number of ways that it could happen. So you can write, I don't have an equation for one here, and I don't have it up here either.
No,
but
because that is the EM algorithm is developed to deal with those type of situations.
Absolutely. Absolutely. You can do this in survival modeling, for example. This is done all the time, where it's possible for every individual whose data is censored to have a different value of censorship. And in fact, if you think about some of these medical studies where people stop reporting back, they stop reporting back at different times. And that's not a problem at all, actually. In fact, the way it looks, when it's done as a discrete time variable, it looks like it's sort of the same for continuous. For each observation, you say, is it alive or dead? And then there's an indicator that that's an exponent that's either a 0 or a 1. And that's for it's a real measurement or it's a censored measurement. So essentially, what you do is you use those data to fit the model to estimate the likelihood up until the point that where it's censored, and then afterwards you're ignoring it. So yeah, it does work.
Gordon, sorry.
Yes.
I was interested in her question, but my problem was I couldn't hear the question, so I
Okay.
just repeat what.
I was just interested in the fact that you're determining the distribution and you're assuming a parametric distribution. And you're assuming that the data that's below the detection limit are following that distribution, but you don't have any data on that.
Yes, exactly. You don't. So your alternatives, you can use Kaplan-Meier. I mean, Kaplan-Meier is pure non-parametric. The limitation of Kaplan-Meier, the big limitation is that it's often not. It doesn't typically have as much power, and you will run into trouble if you have. You will run into trouble more quickly if you have very much censorship. But I mean, I should say, I work often on survival-like problems, and in those problems, I use Kaplan-Meier just as my first look. I always do it. It's the thing that allows you to draw that curve and say that it looks like that instead of, like that. And you can say, all right, those are pretty different. And then in many cases I will go on and use a maximum likelihood to estimate a parametric model. But sometimes, actually, if I just have two treatments, and assuming all the things I need to assume, you look at that and say, well, they are different. I may not need to do anything more to, if my only question is, are they different? If I want to estimate anything other than, say, the median survival time, I probably want to use something, a parametric approach. But sure, that can be good enough.
Isn't this related to those problems where different treatments that you have applied, like the Milka project, the Milka as example that you used? Well, there will be more censored observations for those that are in the treatment that cause the lower, phosphorus in that case.
Absolutely.
So they belong to a different. So they actually belong to a different distribution.
They might. They might. So I mean, the study I first encountered this in, it was a survival study. I was in grad school, and I'll simplify it. I was studying these plants, and I was looking at the effect of water availability on the time to flowering. So that's a survival problem. And I knew from working with them that the ones that were watered less flowered later. That was out. So I, did what people do. I did an ANOVA. Didn't think about distributions. Didn't do any exploratory data analysis. I did an ANOVA, and it was significant, and I was very happy. And then the next day, I looked at the coefficient, and they were the wrong sign. And at first I thought, well, I just coded it wrong, and then no. And then I finally thought about it, and I realized that, well, the ones that water less die a lot more in this particular experiment, and in the real system that I was studying, actually. And what that means is the individuals who manage to flower do flower later, but most individuals never get to flower. Most of them are censored. And I was just chucking them out of the data set, and I was getting the wrong answer from this whole process. And finally, I thought about it, and it's one of those moments in grad school. I thought about it. I finally thought, this must come up all the time in medical studies. And I went to the library, and I discovered that there were shelves and shelves of books on this sort of stuff, and it was all pretty intimidating because there was lots of basic mathematical statistics in there. But I quickly learned, well, this is actually a real approachable problem, and I can do it right. And that sort of thing happens all the time. That we get, you actually can get something that tells you two treatments are different, but they're in the wrong. But A is greater than B when really you should know better. It's not. It's just that you've thrown out all your censored data. Okay, so we can also. So I'll warn you. There's terrible terminology in this area. So there's such a thing as multiply censored data, which is so multiply censored is exactly where things are censored at different values. So some things are now censored at 0.5, and some things are censored somewhere else. And what I've done, I have a script that generates a second data set, and I guess when we run it, initially it winds up giving us, for the first few data points, it gives us the same values. But I guarantee you that there's more than one value in there. We can take a quick look, and probably should, at that.
So you mean like if you have two different people measuring it, so you have two different limits of detection? Is that the idea?
Well,
something like that, where you'd have two different.
Or there are days when your machine calibrates differently, or you drop out of the study. You stop going back to the doctor after 6 months, and I stop going back after 8 months. So that's exactly the problem. It's multiply censored. So we can do the point is we can do the same thing. Oops. We can handle multiple censorship quite easily, and so this NADA routine does this very nice thing where it will draw a box plot. Here's the box plot for the true uncensored data. And here's the estimated box plot for the censored data. And you can see that they're not the same. And the means are awfully close to the same. For this case, everything below here is censored. So in fact, what NADA does for its box plots is it draws them. It draws this for I think it's the highest value of censorship that it sees. So they're going to be. So in this case, it's multiply censored. So there are some points under here that are uncensored. We can't see that. We need a fancier routine, and nobody has written it, and I'm not going to. But the point is, that's actually a pretty good estimate. There's no doubt about it. This is missing the very low values, clearly. And if that was our interest, we might need a different approach or better data. But for most of the things that people are interested in much of the time, this has done a fairly decent distribution, job of describing the data. Okay, so how do we get some insight on censored data? So first of all, I told you there's terrible terminology here. There's a whole. You can write a page glossary, maybe 2 pages of terminology that people use, and it's not. And like most such things, it's not all consistent. And so I'll just warn you that, and I'll say some bits of it you can ignore for now. And let me just introduce a couple of the terms. First of all, so censored data are the case where, so a censored data value is the case where all we know about the value is an inequality. It's not y equals 7. It's y is less than 8, or greater than 50, or y is between 10 and 20. That occurs too. So left censored is the case where it's below something. Right censored is the case where it's greater than something. And interval censored is, where it's between 2 values. So let me just give you an idea. We've talked already about left and right censorship. Interval censorship occurs. So a good example comes from my field, although it can come from many others. So if I'm going out to a field study, let's say I go out every week. I'm trying to determine when the leaves come out on some tree. So I go out every week and I look and say yes, no for each of my sample trees. And then, well, there are 2 possibilities, but let's assume every week is adequate, or even we could say every day. Then I get sick and I get the flu, and I can't go out for 3 weeks. Do I throw all my data? And I go out, and a lot of the trees have leafed out. What we know is they leafed out between when I was last here and today. It's certainly not, and that's the best we can say about them, in fact. So it's interval censored. Now, I want to caution you. You can start chopping very finely here, and you can say, well, I never know. I said y equals 7, but I'm using, let's say, a ruler. It's 7 centimeters. Well, of course, it isn't exactly 7 centimeters. It's something. If you had a better instrument and you paid more attention to it, you'd get a somewhat different reading. But there is the point at which you start making yourself insane, and you start convincing yourself to use methods that are completely unnecessary. If I'm studying trees leafing out, I guarantee you, as a biologist, every day is often enough. The worst I'm going to be off is by a day. But if it's just not it's not a problem where I need to know the second they leaf out, and in fact, it's not definable either. And similarly, there are people who will say these things like, well, I went out every day. I was asked to review a paper where somebody was doing that very thing. And they said, I went out every day, and one day no leaves, the next day leaves. So it's interval censored, right? Because I don't know the hour. Well, What would be the point? What would that tell you? How would it change your answer? How would it change your insight into the science? There are points where you can be silly about this. Okay. Truncated data is a somewhat different problem. So censored data is, data points are censored. I don't know your height exactly. I know that you are taller than something. Data sets can be truncated. Data sets, truncation refers to the case where I don't even know about data points lower than some value. Typically from some design reason, I have no ability to measure that at all, to even be aware of their existence. So they are somewhat different problems. My big point here is, if I can work on this stuff, you can too. It's not, just, well, like I say here, take a deep breath. You can cope with it. There's a lot of terminology, and yes, people don't use it all entirely consistently. Although some things like left and right and interval censored, I've never heard anybody use that to mean anything other than what I just said. All right, so I just said this. These are the definitions of left, right, and interval censorship, and it's a little bit clearer, and you have it in writing. All right. A data set might include all of those. You can have some data points that are left, some that are right, some that are interval censored. It's a pain in the butt to do the analysis, but of course, in principle, you can do the analyses. Censorship can come from your design. For example, the technique you use for measurement. Your machine only measures with down to some limit or up to some limit. Very often, that you have to stop measuring after some time. Like I said, your funding runs out, or they hand you your PhD, and you say, I'm not working on this anymore. Or actually, there are many designs, including in medicine and a lot in engineering reliability studies, where it's deliberately designed. I will stop measuring after so many events. Once we're going to give this drug to 100 people, and after 10 deaths, we will stop giving the drug. You can also get censorship from random factors. This is usually what's called loss to follow-up. It's when the patient stops coming back. In my sort of study, you put tags on trees, and sometimes they fall off. Or you accidentally kill one of your study individuals. It's also a source of random loss. And as I said, you can have multiple censorship values. Okay. So what are some of these? Let's just make sure we understand. So let's look. I have a list of a few of these. So we're measuring a concentration of pollutant in water, and our machine only measures think quantities greater than 5 parts per million. What is that? What's the? We're in terminology camp now. Right censored? How many think right censored? How many think left? Okay. It's left censored. It's things lower than 5. Things 5 or lower, you get a measurement of 0. Okay. So I've read studies where people do this. We're estimating the distance that seeds disperse from a tree. Or actually, I know a study where people were actually looking at the aerodynamic qualities of seeds, and they built a tower and they'd hold a seed up, and they had a measurement of wind. It was all and they had aerodynamic models, and they'd watch how far it went. If you think about it, there's a problem, which is you can only see a seed so far, right? Once it's far enough away, you have no ability to see it anyway. And that will vary with how dark it is. It might depend, if you're doing it visually, it might depend on when you blink, or how, the fact that you're sleepy and you're not paying as much attention that day. There's the point at which it disappears to you, is the point. So what's that? What's this case? Yeah, this is right censorship, exactly. Well, the example I keep mentioning, we're estimating survival and some of our individuals die because we step on them. And we're not interested in that as a mechanism of survival. You know? What's going on there?
Multiple censored.
Oh, well, yeah, it's probably multiple censored, probably, unless you step on them all at the same time. What else is it?
Random.
It's random.
Same way.
It's random. You hope. Unless you're picking out the ones you don't like.
Also left censored because you're killing them off before they would otherwise die.
You're killing them?
Before they would otherwise die. So is that left censored?
No, it's right censored. You know that they lived until you stepped on them.
Okay. But it could have been, okay.
Right. Yeah, so it's random. It's right censored. Yeah, it's almost certainly multiply censored. Or in my example, well, we've already talked about something just like you're estimating the time at which plants flower, and you go check individuals every week, but for some reason your car breaks, and there's a month when you can't get out there, and so what's that?
Interval.
Yeah, it's a case of interval censorship. And there might, obviously in that same study, there could easily be right censorship as well. There could be individuals that die before they ever flower, as an example, or you could step on them too. Okay, so let's talk a bit about regression approaches. So I'm going to do a little bit more simulation here. And what I'm going to do is we'll again assume a log normal distribution, and we'll use ad hoc simulations. And what I'm really doing here is using much larger data sets so that we can so that regressions actually have a chance of working. And so here are what the distributions of these look like. So this is the original log normal distribution. And you can see that it's nice and symmetrical. And here's what it looks like when we set censored values to the cutoff point. Well, when we set the censored values to half the cutoff point, we've done this very odd thing, which is introduce this enormous bar here. And when we set the censored values to 0, it's also pretty. For certain purposes, this might look. You might get away with calling that something close to normal. People do it all the time. Mm-hmm. And for other purposes, it's clearly not. Okay. So let's do least squares regression with these and ask how well it works. And so I'm going to ask. So I've also, so I'm going to ask. I'm going to fit each a linear regression model to each of those, so for the uncensored case, I should get for this particular simulation, I should get a mean of 0, an intercept of 0, and a slope of 0.5. So for the uncensored case, it's awfully close to 0, and it's awfully close to 5. And my R squared, that might or might not impress you for some, depending on what field you're in. But the point is, it is what it is. I mean, there's a fair bit of scatter in these data. Okay. For the case where I've censored and I've just arbitrarily substituted the limit of detection, my mean is way off. Instead of 0, it's 0.22. My slope is not nearly as badly off, but that's an underestimate. It's 0.42 instead of 0.5. And, well, my R squared is actually pretty good. Don't rely on R squared to tell you whether you have a decent model or not. When I set it to half the limit of detection, I've actually got a pretty good estimate of the intercept. I'd be happy with getting that close for most things. And I've got a not too bad estimate of the slope either. In fact, it's not too bad. It's quite good. Yeah, it should be 0.5. It's 0.49. I wouldn't complain about that. You notice that my R-squared isn't any better. It's not. Again, don't. And when I set it to 0, when I just say everything that I measure is 0 is truly 0, instead of an intercept of 0, I get an intercept of -2.
Yikes.
It's wildly off. Instead of a slope of 0.5, I get a slope of 1 1/4. And I should say the R-squared, it's lower, but it's not vastly lower. It's a horrible model, in other words, and yet, yeah, you know, it's the line is not too far from a number of the points. And I think, right, so here are the fits. This is the true. This is actually the this is the true fit, and this is the case where I've set all the censored data to half the censorship value. This is the case where I've set them to the censored value, and here's the case where I've set them to 0. So you can see, I mean, some of these, I mean, that is a pretty good fit.
Wait, so I'm sorry, I might be slow here, but the no censoring data, how do you do that?
Oh, because it's a simulation, I know the answer.
The actual value.
Yeah, no censoring is I'm fitting this on the uncensored value, fitting it to the uncensored values.
So those are 3 different options that people would use if they were below detection or...
Yeah. And so for these data, this one actually works pretty well.
Yeah.
It doesn't necessarily, there's no guarantee that it will, but it happens to work pretty well here. This one works more poorly, but it's not really all that bad for many purposes, depending on what you want to know. That one works quite poorly. I mean, I suppose what we can say is, well, at least it preserves the fact that the sign of the slope.
But the thing is, in a real case, you wouldn't know.
In a real case, you wouldn't know. Yes.
You need to choose the best option.
Exactly. Exactly. I should say one other thing. So in this graphic, the large squares up here. This is the limit of detection is right about here. So the graphic is drawn so that these are darker. The uncensored values are darker, and the censored values are grayer.
So it seems you left out the other option of just throwing out the data that you don't have information for.
Oh.
Or-
Yeah, I did leave it. That's because I just sort of said, don't even think about it. we could fit. I mean, that wouldn't be hard to do. Okay. We could just say, we could take a subset of the data and say, ignore it if that indicator is true, and refit it again. One of the difficulties with that is now we also have a different sample size, and so knowing if you're comparing them, I mean, you could say it gives me a good or a bad fit.
But do you think even the original fit is really a good fit?
No, it's the best fit. It's the true fit, in the sense that it's the true.
You see, what you have is the data which
With a lot of
is
scatter.
sparse
Yeah.
in the two ends. So therefore, I mean, if I just look at it, the linearity comes because of the sparse data. But otherwise, it looks like a random data.
Well, it is random. I mean, it's random. It's random.
No linear relationship.
No, there is a linear. No, I simulated a linear relationship with large variance.
Yeah.
And so, yeah, so of course there's going to be a lot of scatter here,
Yeah,
because it's large variance.
If I look at the entire data, it looks like your zero model probably is a little closer to the actual linearity than the...
Well, part of the problem is that there are points overlying each other here, so you can't see them all.
But you know
But-
the solid line is a linear model because you defined it.
Yeah, but that data doesn't look... I mean, if you are going to fit a
Okay.
model, that's not going to be that sort.
Okay, but let me try to convince you by opening the R code that simulated that. So it is that one. Okay.
No, I think you can continue because I don't want you to hold up.
Okay, so now it's actually pretty simple. So so this is 500 log normal points with a mean of 1 and a standard deviation of 1.5. So it's very large variance. So it's not surprising that there's enormous scatter. I mean, no linear model will account for that. And obviously, and that's for x, and then for y, it's y is simulated as half of x plus a normal with large variance as well.
So it's basically what you have is log linear.
Yeah, and it's linear on a log scale, yes, but it's huge. It's huge variant.
Yeah,
But
that's why that discrepancy in your figure and
Yeah, that makes sense. So if you fit a
well,
log model,
no.
it's the same.
This is on log scale. I'd have to check the code to see whether this is log scale or not. In any case, the point is sometimes ad hoc substitutions can work, but there's absolutely no guarantee that they will. There's no theoretical reason why they should. See what I
Well, I just wanted to point out that I find this as a very interesting example because usually in real data you have a huge scatter.
Mm-hmm.
It's rare that you have a regression that is right on the line or nearly there. Yet the thing is, how can we estimate best what we're trying to estimate, even if data is very variable? If you had an example where you had little scatter, I think it would be me that would raise my hand and say, look, but how is this going to work in the real world
Yeah.
that is so scattered? So I'm interested in this kind of example. Because
Yeah. This also depends on the nature of the problems you're working with. And the kinds of problems that Simonetta or I work with, if everything was close to the data, to the line, you'd probably be accused of faking it. It happens. I've done some where it does happen. But they're usually very simple experiments where you already know a lot or where you've done a huge amount of rescaling. But it's not rare at all for many things, certainly many environmental sorts of data, but I would think many business sorts of data as well, to get enormous scatter, because just there's a lot going on out there. And I mean the other thing is the other difficulty is that people fit models and then they'll say, you see, if I've explained it's significant or it's the best fit. Yeah, well, it's the best fit of the ones you tried. And there's obviously also the big question, is that sort of model what you want? Perhaps for some circumstances, of course, you want to describe what the cloud looks like.
maybe quadratic.
Pardon?
Maybe quadratic.
I don't think, well, in some, yes, exactly. It could be. In this case, we only tried linear. I'm pretty sure quadratic wouldn't work here, but of course.
I have another question that is just out of interest. Why did you choose to simulate the log model? Is there a reason? Just because it could have been.
Because I felt that way the day I wrote. Yeah, it could have been anything. Yeah.
But I think also because much environmental data is been found to be log normally distributed because it's the many nonlinear processes all interacting to create the concentration. So I mean that's a standard transformation.
It's pretty common in I'm sure in the sort of things you work in, or the sorts of things I work on, where there's that, or you just have these things where we're looking at things like the sizes of organisms, where, oh sure, it's often log normally distributed.
And the explanation I've heard for that is just basically that because you have all these processes operating in many different scales and they're not all nonlinear processes, it leads to a log normally distributed. So concentrations are typically log normally distributed in nature.
Yeah, I'd agree with that. I mean, there are some things, for things like survival times, for certain kinds of processes, they're more often gamma distributed, for instance. But that's because it's because there you're looking at time until an event rather than the measurement of something. But sure.
Can I ask you a little question
Absolutely.
that I never quite get? All right, so for your problem where you said I assumed it was 0, the concentration was 0, or 10 to the 0 equals the constant, because you say the log of 0. I mean, you can't do log of 0.
Ah, yes.
So,
yes.
I'm just curious.
Oh, let me show you.
Because that looks like it was like a scaled data that then you set it
Oh.
equal to 0, which make it a mean. Like
Yes.
the deviation
So
of 0.
so let's look at when we look at the residuals from these things, you'll see what happens. And in fact, what happens when you look at the residual plots is you'll say, oh, so these arbitrary assumptions really are arbitrary. And it becomes, in fact, setting things to 0. The problem is they can't be 0 because you can't take a log, so then you have to either throw them out or say, well, let's call it 10 to the negative 6th. And that turns out to really affect your statistics.
That's what I was
Yes.
thinking. I was wondering.
Okay, this is actually, I actually really like. So I said before that making these ad hoc substitutions creates new patterns of variation in the data that aren't there. So that's what I want to show you now. So yeah, exactly. I'll pay you later for asking that question. We can't plot the case where we set y to 0. So typically we say, all right, it's something really small. And so I do that here. And so now we're looking at the scatter plots. And we have square is uncensored and round is censored. So here's the original data set. And you can see that there's a linear core to it, but there's a lot of scatter. When we set censored values to the censorship cutoff point, you can see we introduce a straight line there, which is fairly bizarre. When we say, well, let's make it, better estimate would be halfway between that censored value and 0. We introduce a straight line there with a break. And when we say, well, let's just do that, we have a straight line with a big break. And so, I mean, think about it. So what you've been trying to do by using these ad hoc substitutions, here's your real data set. As Simonetta said, the problem is, of course, you don't actually know. You don't know what these values really are. But what you've done by these ad hoc substitutions, which are really natural, it's a real natural thing to say, well, I don't know, call it 0 or call it something, is you've changed the shape of the data set. And so it's no surprise that when you then go to fit it, you get answers that sometimes work pretty well and sometimes, like in this one, work very poorly. That's why that regression line looked like this. When we look at the residuals, so we have an appendix in the book chapter that discusses this. There's an appendix to the chapter I will try to remember to put. It's online, PDF with some R scripts. I'll try to remember to put it in this folder so everybody can have it. And I went through the exercise. I was curious about it. Of, well, what if I keep setting it to smaller and smaller, and just looking at sort of how badly it changes things, and it's horrible. It just says, there is this idea. Well, I'll approximate 0 more closely, and in fact, you're often making things worse, which is really once I thought about it, I realized that of course that had to be true, but it's annoying because it seems like it shouldn't be the case.
The other thing seen people talk about doing is, I'm saying that this is a better way to approach this, which is basically to sample for those values between 0 and the cutoff.
Mm-hmm.
And I always say, well, what do you mean by sample? Sample what parametric distribution are you assuming? And so you're saying the great way to do it is to do it based on the QQ plot of the whole distribution.
Mm-hmm. So that's one way to do it, yes.
You know, because you can't
Yes.
just say, okay, if you have a log normally distributed value, what are you sampling? I mean,
Yes. Yes, right. You have to fit it to something.
Right.
Exactly. No, I think you're exactly right. So if we plot the residuals, you can see just how bad the situation is. This is the no censoring case, and I mean, that, you know, linear regression there should work. This is the setting it to the cutoff point, and if you got that as a residual plot, you should be worried. And if you got these, you should wonder what the hell is going on with your data. It's too bizarre, you know. And the fact is, I'm not sure I've done all of these, but I've done some of these in the past. And you know, I confess my sins now. This is a bad way of approaching things.
So even the CO2. So you're going to give us the answer. Right?
Yes, the answer, yes.
You have me convinced
Well,
that dividing by 2 is maybe the way to go.
Well,
now, not so much.
well, it's, you know, it worked in this case, and it sometimes, and of course, if you think about it, it is a better answer. It's just it's not without its problems, and you can sort of see that, right, there can be some real problems with it.
But only if you,
I mean, how bad is this? There is no way to say this is really bad, but the point is, it's suspicious. It's something to worry about.
Did you do some sort of formal normality tests on these residuals? Just curious to see how far they are from what?
Oh, to do a test for equality of variances or something like that? You certainly could. And
I think more appropriate is to do a test of randomness.
actually, you're right. In fact, usually, don't most of those tests ask for sequential points and ask whether they're correlated with each other? Like whether you get ups and downs or something like that?
But in the regression case, it's not sequential.
Okay. Well, okay. So, right. This is highly non-random. I think you're right, Ram. I mean, that's terrible. That's just terrible. This is pretty bad. And, you know, well, they're all clustered, but there's obviously a straight line that you've introduced, which is sort of a problem. Okay. So, we can also another way of doing this is to look at the I did have a probability plot in here, I just didn't remember where it is. We can look at the probability plots, and, you know, we should be able to see that, so here's the case where we've set it to 0. And you can see that that's so far from the straight line we would like, that we shouldn't be doing this. We shouldn't be fitting that model. It's probably a very bad idea. I think this is the only one I have. I didn't fit it. I didn't do a probability plot for the others, but the code's in the R code, and you could easily take the code and use it for the other data set.
Is that some kind of confidence interval?
Yeah, this is I think a 95% confidence interval around. Yes, it's a 95. This curve is most of the time outside its own 95% confidence. Right. Yeah.
Okay.
Yeah. Yeah. No, this is. Yeah. Don't do it. Right. Okay. So I mentioned before this distinction between bias and consistency. So an unbiased estimator, we all learn about things are biased. So if something is unbiased, on average it estimates the true value. So it's a property of small samples. So you know, if I have a normal data set, my linear regression will on average get me the right slope and intercept. Not saying it's always right, it's on average right. Consistency refers to limits when you get very large data sets. So as we increase the sample size towards infinity, a consistent estimator will make the estimates actually approach the true value of the population quantity. So if we now sample 10 million individuals for their size, and the relationship between their size and weight, let's say, our estimate, as it gets larger and larger, will approach the true value, not just on average, but the true value. So it's a property of large samples. So if we use these ad hoc substitutions, we can get inconsistent estimates. That means that no matter how big our sample size is, we're not going to be approaching the correct answers. So in certain ways, it can be, this sounds worse than bias, and it can be, a biased estimator, as you get to bigger and bigger samples, can still give you a good answer. It's just that with small samples, it won't. Big enough sample, the problem can disappear. An inconsistent estimator you can't trust at all, is essentially what I'm saying. And these ad hoc substitutions make our estimates inconsistent. Okay. So the bottom line, don't use ad hoc substitutions. Now, I mean, there is the obvious question, what if only one of those data points was censored? And I think my answer is, well, if only one were censored, what was that, 500 data points? If only one was censored, no matter what you do, it's not going to matter very much. I'm not trying to encourage you to use bad statistical practice, but you know, like everything, I mean, all of these cases where you're fitting models, these aren't usually the true model. These are always exercises where you're approximating and making assumptions. And you know, sometimes you can make assumptions. If you have a tiny bit of censorship, it may well not affect your outcome. And so I'm not telling you, you know, like I was telling you about the guy who said, you know, there's this interval in which I don't know what the plant was doing. That's just being silly. You know, just use ordinary statistics and don't worry about it for that sort of case. And you know, I don't know what to tell you. If you have one censored data value in a data set of 500, should you leave it in or throw it out? You know, what should you do with it? I don't know, but I guess my point is, then it probably doesn't matter. Whatever you do with it will probably not affect your result. And in fact, a way to approach that, and I've done this, is do several things, several sort of limiting things, and say, it doesn't change my answer in any qualitative way, so fine. Then it's defensible. But don't go ahead and do this in general and assume that it's okay, because it's generally not. So let's move on and talk a bit more about regression. So now I want to generate a larger data set so we can use some of the parametric regression models easily. So that's all this does, is it generates a large data set. And there's a model that's used really heavily in some environmental sampling and some social sciences, and it's called Tobit regression. It's the guy who invented it was named Tobin, and it's sort of conceptually related to probit regression, and so some funny person named it Tobit. It wasn't Tobin, actually. And so here's the idea for Tobit regression. So for the moment, assume that we have a linear regression, and what we're going to do is model what we observe. So the idea here is we have a linear model if we're above the censorship level, and if we're below the censorship model, level we observe censored value, right? And I'm sorry, one bit of notation. Y star is the true value, and Y is what we observe. Okay. So then we can actually write out the expectation of Y given X, and it's essentially It's this. It's the probability that we have an uncensored value given x times the expectation of the normal expectation of y given x plus the probability that we have a censored value times the limit of detection. So it's actually sensible. This takes a minute to run through, and don't sweat it too much now. You can go through it at your leisure. But that's part of the reason of sharing these files with you. But it's actually quite sensible, I assert. So we can actually write down a likelihood of this. And so what we do is this. We use the cumulative distribution function to estimate the probability that the point is going to be censored, and the probability distribution to estimate the probability of y given that it's not censored. So we're sort of fitting two pieces. We're taking the part that's censored and doing one thing to that, and the part that's uncensored and doing something else. That makes sense? Sort of? In a roughly intuitive way, not thinking about the math, just sort of squinting your eyes. And okay. So here's an example. So what I've done here is this is the same data set, but quite a bit larger sample so that all of this works really smoothly. So again, everything above this line is uncensored. And what these are, so these are the probability distributions at each of these three values of x. And you can see, well, up here we can get a very good estimate of the probabilities of x. And here it's not as good, and here it's terrible, right? We can't really estimate this. If that was all we had, we couldn't estimate it, really. But but at each of those values, we also have, well, what's the proportion of our samples that are censored? Well, when x is very small, almost the entire, is that actually 0? No, almost the entire sample is censored. When x is in the middle, remarkably close to half of it's censored. And when it's really large, hardly any of it's censored. So we can actually get fairly decent estimates of both that PDF and that CDF. And we can fit the censored regression model. What I'll tell you about doing it in R is this. NADA will do, you can use the NADA library, but it's actually, the NADA library just calls the survival library anyway. And if you call the survival library directly, you can access more information from the output. So I would recommend just doing that. So that's what this does. We are fitting a, I'm going to guess that nobody can read this.
I can.
Good guess, huh? You can read, yeah, Christina can read it. All right, let me make that a little larger. That's really small. So just to explain some of what this does, oops. So the serve-reg routine in the survival library, what we're doing is we're saying, okay, I want you to fit, something I didn't tell you, it's crucial. So when you fit survival type data sets, you're fitting an object, and I'll call it this. Let me see if I've written that incorrectly. I have. Okay. What you're saying is, instead of one value, for each data value, you actually have two quantities. For each data point, you have two quantities. You have, if it was a true survival problem, you'd have the day on which you're saying it's dead, and whether that's a censored value or not. So it's always got to have two values: the data value and the indicator as to whether it's censored or not. And then it's going to equal some stuff. I'll say AX1 plus BX2 plus a bunch of stuff and some error. And I don't even necessarily mean that has to be normal, or it's whatever distribution it is. Okay. But that's the important thing. So in survival problems in R, there's this function called serve. And serve sticks those quantities together in a single object. That's all it does. This is one that, unless you really want to know about the details of programming, you don't really need. It's not that it's doing a lot of fancy mathematics. It's mostly doing that. So all we're doing is we're saying, okay, we want this value. Here we're taking the cases where the censorship indicator has the value of 1. We're telling it that you don't necessarily have to, that it's left censored, type equals left. And then we're just saying fitted as a function of x. And we're going on to say that the distribution is log Gaussian, log normal. The default there is exponential, but that's the default distribution. So you want to specify one because exponential is usually not what you really want. And then we can get a summary for this regression model. And in this particular case, it's giving us, you remember, it's the same, this is the same data set. Intercept of 0, it's estimating it as 0.02. Slope of 0.5, it happens to get 0.5 or almost 0.5. And there's a scale estimator, which is well, it's related to a variance estimate. I'll just say that it's playing a role in this model, something like variance does in a normal model. And so we've got a fairly decent fit from this model of the same data. Anybody have questions? So let me go back, just remind you what we've just fit the data to. This is the case where we've fit, we have this relationship between x and y. We've got around half the data are censored, and with a single cutoff. And we've used two things. We've used what's the distribution of the data when it's uncensored, the actual PDF of that distribution, the probability distribution function, and we've used the probability that the data point is censored down here. So those are combined in this model to allow us to make this estimate. Okay. And here's the plot of the regression, the fit. Here's the linear regression on the complete data set, and you can't really see it. You may be able to see it on your individual screens, but the little dots are the fitted model using the censored regression. And that's pretty good. It's more than pretty good. It's great. Obviously, this is a really simple case, and I simulated a lot of data points to get it to do this. So your mileage may differ.
So the probability that it's censored, is that predicted from the population of data that you have that are below your cutoff? Is that okay?
Yeah, it's predicted from, in this particular model, it's predicted, we can say, well, at this value of x, the probability of the data are censored is nearly 1.
Okay.
And here it's, what's that, 0.45.
Yeah.
And over here it's nearly 0.
So does your code walk through those three possibilities, or
Yeah.
I'm missing that?
Actually, the code draws all these graphs.
Okay.
But no, the code draws the graphs, and it calls those routines that other people wrote that actually fit the model. But it does do this part. In other words, what I've done is to sort of, I've used their canned routine and I've drawn some graphs to sort of explicate it.
Right.
But I haven't done, I'm not going to write the code that does the integration. Other people have.
But somehow it walks across what your values of X are and calculates.
Well, it takes, yes, exactly. It just says at each value of X, at all values of X, what's the probability that the data that are censored? Yeah.
I see.
But, so for the real data set, you'd only have those dark circles. And so
No. No, well,
for
what you would have is you can't draw it. You'd have this, but with no scale there because you don't know what the values are. You just know that they're below here. But you'd be able to still say, I know that there are 78 censored values at this value of X, and I know that there's 190.
Right. Don't know where they fall.
Yeah, you don't know where they fall exactly. Right.
Okay, so you basically know the number that are below.
Yeah.
Let's do a simple left censoring.
Yes.
One,
Yes.
One
Yeah.
MDL. You know how many are below that, how many measure below that. And then
Yeah,
and then you have all the ones above it.
But since we've assumed a linear model and normality, we can estimate.
Okay, so
That's
what you do. So
we're-
you're basically
yes,
saying,
we are
okay,
modeling
I'm
it.
assuming a linear model, and I know it's going to be,
Yes, exactly.
So because of that, I know that at the MDL, the probability it's censored is about about 0.5, and
Exactly.
okay.
Yeah. I mean, it's definitely a little bit of trickery, or it's
No. I
trickery.
just wanted to make
It's-
sure I understood.
Yeah.
What's being.
No, I mean it's based on some assumptions.
No, actually, when you are fitting a log linear model,
Mm-hmm.
the actual relationship between X and actual Y is exponential.
Is a what?
Exponential, right?
Yes.
So it's not actually linear.
No. But you've done a linear you've log transformed it. Yeah.
So it's actually an exponential model relationship.
Mm-hmm. Absolutely. Okay. So let me just say a few things about truncated data. So there are a large number of things you can do with let me finish by saying this, large number of other things you can do with censored data. As I said, mostly I work with survival sorts of problems, but we clearly get censored data in lots of other settings, and this is just an introduction. So for truncated data, we get truncated data when our design is such that over some range, we're not even sampling. So censored data, I've sampled something, but for one reason or another, I don't know the true value. I just know an inequality about it. In truncated data sets, I don't actually, I haven't even sampled over part of my distribution. So here's two examples. One is I'm interested in studying the spread of a weed species. And I have remotely sensed data that has a resolution of 30 meters. Pretty standard for a lot of remote sensing. So there's some range over which, so as I'm fitting models to the spread of this pest, I have to confront the fact that over certain scales, I have no idea whether it's there or not. It's not that I know it's there, but I don't know how fast it moved or something like that. I really can't tell. Another case is this one where, let's imagine we're trying to estimate the diversity of phytoplankton, and I have a net that has fairly large pores. So I scoop it up and I say, well, how many species are in here? And I miss all the little ones. It's not censored. I don't even know about them. There's no way I could have sampled them. Well, I could have got a better net, but I didn't have a better net. This comes up in a lot of disease settings where the studies are, they'll study things like the effect of a treatment on people once they go to the hospital. And so you don't know anything about all the people who've never gone to the hospital, or who don't go until the tumor covers most of their torso, or something like that. And so you can see that there are real problems with us looking at data of this sort. We can have, again, data that are left or right truncated. That is where we don't know about the small things, or we don't know about the big things. But this is now a property of the entire data set instead of individuals. Remember, censorship, there are individuals that left your study for one reason or another. In truncated data, our design means that we don't even know about, say, the left tail of the distribution at all. And so a lot of the ideas are related, and a lot of the terminology is similar, but it gets confusing because it's not the same problem. And again, ignoring truncation can lead to inconsistent estimates. So what I do in this code is, I remember when I got to this point, I thought I was really tired of programming. And now I've got a program truncated data set. Wait a minute. It's really easy. All I have to do is say, ignore the left-hand side, or the right-hand side. And by the way, I should tell you that routines like nada, what they do, for certain things like the Kaplan-Meier estimate, that's always it's survival estimate. It's non-increasing. All they do is reverse the problem to you. So if you have right-censored data, the code just reverses the problem and estimates it that way. It's actually kind of clever. So here what I do is I use my simulated data and just take the part of it that's greater than the limit of detection, and then to make sure I had a sample of the same size, I take a random sample of the untruncated data set, so I'm comparing comparable size data sets. And
so you're saying that basically the issue of values below limit of detection is really a truncated data problem. We've been talking about it as a censored data problem, but
no. So if I don't know your weight because of the scale problem, I know about the value, and I know that you exist and that you got on the scale, and I know that you are smaller than whatever. If with truncated data, it's if I cannot see people with blonde hair, and so you're just never in my study, and I don't know about it at all. So I've actually chopped off. I've chopped off a tail of the distribution, and the difficulty is I've chopped it off, and then I have to think about how to do my modeling and estimation because I no longer have the distribution that I thought I had, or that I hoped I had in many cases. So so we can't use methods like the regression on order statistics because we don't even know about these data points or Kaplan-Meier. But there are methods, and these are that are similar to censored regression, and what they do is they rely on thinking about how the probability distribution changes from truncation. So here's a maybe too complicated figure for a talk, and here's the idea. We had a normal distribution here, and it's truncated at this point. So all the gray over here, I don't know about that stuff. And what I'm sampling from is the white. But if you think about it, well, what's happened is, in fact, I've made in my new distribution. My new distribution is no longer this shape. It's now I have to take all the. Put it this way. It's this shape, but now I have to rescale everything because it all has to sum to 1. It's got to integrate to 1 for the area of under this curve, right? So when I do that, I get, in fact, a distribution that's this shape that runs along the axis here, goes straight up, and now it's shaped like that. So what I have is a truncated distribution. The mean of that truncated distribution is larger. The variance is smaller because I've chopped off a whole end of it, and I've made more things near the middle. Does that make sense? It's sort of weird.
There is entire PhD dissertation written in the department on the truncated model. So
It's not trivial stuff. It's some of it's sort of difficult. And when I say something, I'm the first to admit I am not a statistician. Yeah, I think a lot of this stuff is fairly difficult, actually. So we need to do a lot of things a little bit differently. And let me just say, so we can still, once we admit to ourselves that this is a truncated normal, instead of just truncating it and saying, it's good enough. Yeah, there's some negative values, but I'm going to ignore them. We actually can write out a likelihood function. Probably not in every case, as a guess. But we can. And there is an R library called Trunc-Reg that makes the appropriate adjustments. So just to show you how this is done, so this is more or less the same sort of fit. We're fitting something that looks like a linear model, y or log y as a function of x, and I'm telling it that it's truncated on the left-hand side, and I'm also going to fit an ordinary linear regression to the model so that we can see that you shouldn't do that. And I'm going to do it for the truncated data as well as for the untruncated data. And how well do they perform? Well, here's the full data set without any truncation, and again, it's the same model. The truncated regression does pretty well, very well, in fact. And the case where we do ordinary linear regression, OLS, on the truncated data, it's not so good. It's off, no question about it. Diagnostics with these models are difficult. Part of it is that the residuals have to be not what you expect them to be for a normal model. I mean, after all, you've chopped off the left end of a distribution, and you've made that peak really high and things like that. So you can't do all of the same diagnostics, and it's no longer a linear model. And so one of the things that people do, and that I think is a useful thing to do, is something like bootstrapping the confidence intervals. And again, I will remember, somebody remind, it shouldn't be your responsibility, but I'll ask you, remind me, put the appendix online so you can get it. Actually, it's already online on our site for the book. Actually, yeah, right. It's all right, but I'll copy it to. Anyway, we need to make sure you can get it. Let me just say, so truncation. One of the places truncation is common is in survival studies. So, for example, people study total lifetime reproductive output, or what's often called reproductive success. And they say, I'm going to ignore the little individuals because the little individuals don't reproduce. But then if you think about it, what you're estimating is lifetime reproductive success of individuals who are already big enough, not of the average individual in the population. The survival library in R does not handle truncated data, but there's a thing called EHA, Event History Analysis, which is what some social scientists call survival analysis. That does it. And I truncate the data very severely here, and I'm using a large sample size. So I generate the data. I'm going to do it with 2 covariates. There was a reason, and I don't remember why. And this is for a survival model, and I'm simulating it as a Weibull distribution, which is widely used in survival models. I'm running out of time, so I won't tell you anything about it. But it's for survival models, it's often the default. And I'm truncating it. Doing the same thing, create a data set that's the same length, but that I've randomly selected from the full set. And here's the basic fit. So I get very different model fits. When I fit my naive model, that is, where I'm pretending I don't have truncation, I get this is my cumulative distribution. For the truncated fit, I get this curve, and for the untruncated fit, I get a different curve, but it's not nearly as different. So the bottom line here is ignoring truncation can give us really bad parameter estimates in survival models as well. If we ignore the truncation, the 2 parameter estimates, for the covariates, they're both underestimated, and the Weibull scale parameter is estimated at a really large value. And that translates into a big difference in what's called the hazard function. It's essentially the instantaneous risk of an event occurring. So just to finish up, censorship and truncation are common, here I say, in ecological data. They're common in most data. They can really severely affect our estimates. They can make them biased. They can, in fact, make them inconsistent. A fair amount is known about how to handle them. Not everything is known about them, and there's no question the theory and just the computational methods are at an earlier stage than they are for handling uncensored and untruncated data. And I just want to leave you with, again, this point. Well, do we always have to use these methods? That's for you to judge as a scientist and a data analyst. Do I need to do this? Is this a severe enough problem? The answer is sometimes. Quite often it is. More often than you think, but of course not always. Okay. And I'm utterly astonished. I am never this precise. It is exactly the time I said I'd finish. Usually at this point I say, oh, and I've got to skip the last 50 slides. I ran through the last half dozen pretty fast.